Alright, so in the last video we stopped at the fate of pyruvate, where I made you understand that there are four fates of pyruvate. There's the one that pyruvate will form, there's the one that pyruvate will form lactate, right? Then there's one that pyruvate will form acetyl-CoA, right? Then there's one that same pyruvate will form oxaloacetate, right? I already did this in the last video. And lastly, I said pyruvate will form what? Will form alanine. These were the four fates of pyruvate. Now for this video, we want to focus on this one, pyruvate forming acetyl-CoA, and that one usually occurs when oxygen is available. So we have pyruvate, going to form acetyl-CoA. This one usually occurs when what? Oxygen is available. Now the idea here is the enzyme that is involved in converting pyruvate to acetyl-CoA is what we call the PDH complex enzyme. So the enzyme is called PDH complex. So in this video now, the aim of this video is for you to understand the enzymes that make up this PDH complex and how they actually convert pyruvate to acetyl-CoA. Alright, so and how you can regulate that enzyme also. That's what you need to learn in this video. So now I will just give you a scenario, a simple form of it. We have, this is pyruvate, 3 carbon molecule is being converted to acetyl-CoA, which is a what? A 2 carbon molecule with a CoA group. So let me just use this star to represent CoA. Are you there with me? Meaning, in order for we to convert pyruvate to acetyl-CoA, we need to remove one of the carbon. So there will be what? Loss of one of the carbon as what? CO2. That's one of the things that will happen. Then alongside, there will be addition of what? There will be addition of what? What do we add? Of CoA. Coenzyme what? A. Are you there with me? Okay, so there will be addition of coenzyme A to the reaction also. Now, these are the two major things that will happen. You remove CO2 and add coenzyme A. Alongside also, there will be formation of energy molecules. So NAD, meaning there will be removal of hydrogen. NAD will enter the pathway and there will be formation of what? There will be formation of NADH. So NADH. These are the three major things that will happen in the PDH complex. These three major things are the things that will happen in the PDH complex. First, removal of CO2, addition of CoA, formation of energy. Okay, these are the three major things that will happen in this PDH complex reaction. Then once you have formed your acetyl-CoA, acetyl-CoA will now enter the what? The tricarboxylic acid cycle, like the TCA cycle or Krebs cycle. Are you there with me? Alright, so let's now see what are the components of the PDH complex. So you write now component of the PDH complex. Component of PDH complex. Are you there with me? So what are the components of the PDH complex? Now this PDH complex consists of 3 apoenzyme and 5 coenzyme. Consists of what? PDH complex consists of 3 apoenzyme and what? And 5 coenzymes. These are the things that make up the PDH complex. Apoenzyme and coenzyme. 3 apoenzyme and 5 coenzyme. So let's list them now. So now I will first give you the 3 Apo enzyme. Maybe let me just write it like in a cartoon format. So imagine this one is one of the Apo enzyme. Imagine this one, this next one is another Apo enzyme. Then imagine this last one is another Apo enzyme. Are you there with me? So these Apo enzymes, I will just draw them somehow, because they are actually bad guys. They are responsible for converting pyruvate to what? To acetyl-CoA. Are you there with me? Now, this first one here, this enzyme 1, let me call it enzyme 1, then this one enzyme 2, then this one enzyme 3. 3 Apo enzymes. Now, this first one here is what we call pyruvate decarboxylase. Pyruvate decarboxylase. Pyruvate decarboxylase, or you can also call it pyruvate dehydrogenase. Pyruvate dehydrogenase. Okay. Either you call it pyruvate decarboxylase or pyruvate dehydrogenase. Now, from the name, you will know what it will do, right? So, what will it do? This one is responsible for what? For removing the what? So, if for example, this is the pyruvate, the 3 carbon molecule, it is responsible for what? For removing one of them. So, it will remove one of the carbon. So, what you are left with now is just 2 carbon molecule. Are you there with me? You are left with what? 2 carbon molecule. One of the carbon structure, one of the carbon has been removed as what? As CO2. That function is carried out by pyruvate decarboxylase enzyme. That function is carried out by what? Pyruvate decarboxylase enzyme. Now we're left with just 2 carbon. This 2 carbon now is what we now react with this enzyme 2. Now, the function of enzyme 2 is that enzyme 2 is having a particular cofactor. Let me just put it here. Let me add it here. Okay, I already told you that this one is enzyme 2, so let me just erase this so that I can put the cofactor there. Let me take the eyes up a little. Okay. So this is enzyme 2, right? Now, this enzyme 2 has a cofactor. It has 2 coenzymes. The first one is coenzyme A. The second one is lipoic acid. Lipoic acid. Okay. Second one is lipoic acid. LIPOIC. Lipoic acid. Now, with the help of these 2 coenzymes, enzyme 2 is going to carry out this function. So one of the functions is that it is going to attach CoA to this structure. It will attach CoA to this structure, and at the same time it is going to pick 2 hydrogen from the 2 carbons. So what will happen is that if it picks the 2 hydrogen from the 2 carbon and attach coenzyme A, what we will get at the end of the day is this. So we have the 2 carbon plus coenzyme A. So let me just put this, the coenzyme A. So plus coenzyme A. So this is what has formed. So we've already formed acetyl-CoA, but it also removed what? 2 hydrogen. So let me just put the 2 hydrogen here. One here. One here. Are you there with me? So what is the function of carbon 2? It transfers the what? Coenzyme A to this 2 carbon molecule that is left. Is that not so? The name is called the name is a bit long, so I will just take my time and spare for you. So the name is called dihydrolipoyl transacetylase. So it's called what? Dihydrolipoyl transacetylase. Dihydrolipoyl Transacetylase, dihydrolipoid transacetylase, because why is transfer this acetyl group to the CoA. Do you understand? Either way, whether the CoA attached with it or this two acetyl group attached with the CoA, but the main thing is that the function of this enzyme 2 is to add the coenzyme A to these two carbon, which is the acetyl group here. Are you there? But which other thing does it do? It removes two hydrogen also. So that's why we now have dihydrolipoid. So in the name. Why this third enzyme, which enzyme 3, it still has two coenzyme attached with it. It still has two coenzyme attached to it. So let me just put it here. Enzyme 3. It still has two coenzyme attached to it. These two coenzyme are what? They are FAD and NAD. So these are the two coenzymes that are attached to the last apoenzyme. So the two coenzymes attached to the last apoenzyme, FAD and NAD. Now the function of this last coenzyme, okay, let me just tell you the name. The name of this enzyme is similar to the second one. It's called dihydrolipoid dehydrogenase. Dihydrolipoid dehydrogenase. So dihydrolipoid dehydrogenase. Dehydrogenase, there's no space. So that is it. The function of this last enzyme is that it will remove this two hydrogen in this lipoic acid here. So the function of the enzyme is to remove this two hydrogen. Let me use this. It is to remove this two hydrogen here and attach it first to NADH. So we'll now have something like NADH H2. Then later on, this two hydrogen will now transfer again to the NADH and form NADH H2. So initially it was first in FAD, then later on it transfers to NAD. The NAD that is formed will now go to the electron transport chain and give us energy. So the whole idea is this: the first enzyme, which is pyruvate, the carboxylase enzyme, its role is very simple. What does it do? It removes the carbon dioxide for us. Is that not so? Then we are left with two carbon. The role of the second enzyme is to first transfer the coenzyme A to that remaining two carbon. At the same time, remove two hydrogen from the carbon. Okay, while the role of the third enzyme is all about taking that two hydrogen that the second enzyme took from the acetyl. So if you look at the third enzyme, all it was doing was battling with what? With the two hydrogen. First of all, FAD took it, then FAD was not able to go to the electron transport chain, so it gave it to what? To NAD. So that is the whole concept. So if I want to simplify it for you now, all right, I didn't tell you something about this enzyme 1. Enzyme 1 is also having its own cofactor. Enzyme 1 is also having its own cofactor, and that cofactor is called what? So let me erase this. Enzyme 1 is also having its own cofactor. That cofactor is called what? It's called so enzyme 1. So it's just one cofactor that is having. That is called TPP. TPP. Okay. So this TPP here is called thiamine pyrophosphate. So vitamin B1, thiamine pyrophosphate. Okay, thiamine pyrophosphate. Thiamine pyrophosphate. Thiamine pyrophosphate. Pyrophosphate. Okay, so that's the full meaning, TPP. So we've seen the 5 coenzyme and the 3 apoenzyme. So let me summarize their function. So if I want to talk about enzyme 1, enzyme 1 function is very, very clear. It does decarboxylation. It's the one that removes the carbon dioxide. So decarboxylation reaction. Is that not so? How about enzyme 2? What is the function of enzyme 2? The function of enzyme 2 is to transfer the acetyl, to coenzyme A, or if you want to take it the other way around, transfer coenzyme A to the acetyl. But let's just take it from the first side. Transfer what? The acetyl to coenzyme A. So transfer, this is the first function. Transfer the what? The acetyl, which is the 2 carbon molecule. Let me put it like this. So what? To coenzyme A. Then which other function did it do again? It removes 2 hydrogen from the acetyl also. Abi? It also removes 2 hydrogen from the acetyl. Removes 2 hydrogen from the acetyl. From the acetyl group. Is that not so? Then what's the name of the third enzyme? The third enzyme, what did that third enzyme do? The third enzyme, what is the function of the third enzyme? It's all about waiting, taking the hydrogen. So all they did was to dehydrogenate, or they are busy with the hydrogen. Let me just put the word so that you understand. Busy with what? With the hydrogen that the second enzyme took. So the first, who first took the hydrogen? Which coenzyme first took the hydrogen? FAD, right? So FAD first took the hydrogen, so we now form FADH2, right? But FADH2 was not able to go to the electron transport chain, so it transferred that 2 hydrogen to what? To NAD. We now form NADH plus H. This is how it is written. Okay. Then this one will now go to the what? To the electron transport chain. Are you there with me? Okay. So I believe with this now you've understood the 3 coenzyme and 5 coenzyme. So let's just list them. Okay, I already list the enzyme. Let me just list the coenzymes. So the 5 coenzymes, let me list them proper for you. The 5 coenzymes. So the 5 coenzyme, number 1 is what? Thiamine pyrophosphate. Number 2 is what? Number 2 is what? Number 2 is what? Coenzyme A. Number 3 is what? Here's number 3. Lipoic acid. Number 4 is what? FAD. Flavine aldehyde dinucleotide. Flavine adenine dinucleotide. I think that is the full meaning of this. But this is coming from vitamin B2. So the last one is your NAD. Okay, so these are all vitamins. So this one is thiamine, which is vitamin B1. So vitamin B1. So vitamin what? B1. Coenzyme A is from vitamin B5. Pantothenic acid, B5. Lipoic acid, this one is not a vitamin, but NAD is also a vitamin from vitamin, that is flavin, B2. Riboflavin is B2. Then NAD is also a vitamin from what? From vitamin B3. Are you there with me? Which is niacin, NAD, nicotinic acid, niacin. Are you there? Alright, so when you are being asked now to explain the PDH complex, I believe you can explain it properly. You understand? So in the next video, what I will do is to explain to you the regulation of this PDH complex. Then we will go into the Krebs cycle. Okay.